So today's lecture will start from, let me give you some idea of the unit. So you learn all gram positive already, okay? Go back to review. Last week, you learned gram negative cocci. Still remember Neisseria. We have two Neisseria, gonorrhea and meningitis. And also learn some gram-negative coccobacilli. They are Haemophilus, right? And something else. So you forget it. Today, we go to lecture 17. Lecture 17 talk about bacteria in two families, Enterobacteriaceae and Vibrio-Naceae. These two families cause diarrhea, mainly cause diarrhea, but they can cause something else. Meningitis, can sepsis, can make the middle ear inflammation, and also the other organ. But main symptom of bacteria in these two families is diarrhea. The way we classify or identify bacteria that are gram-negative bacilli. So every bacteria you learn today are gram-negative bacilli. We use biochemical tests for differential these bacteria. So let me give some time to talk about the biochem tests that related to microbial media. So the microbial media can be divided into three main types based on the texture of media, the physical state of media. The first one is solid media. Solid media is media that contain 1.5% of agar, or maybe larger than this. Okay, semi-solid media contain less than 1%, or instead of agar, some microbiologists probably use gelatin instead of agar. So for testing gelatinase enzyme. And the last one is liquid media. Liquid media, as you call broth. Okay, no agar contained in that media. Let's say if we classify media in terms of composition, chemical composition, we probably classify media to be synthetic and non-synthetic media. Synthetic media is mean like the media that all chemical in the media, you know the concentration, the correct concentration. For example, synthetic media that use so many chemicals, you can calculate the amount of sulfur, you can calculate amount of magnesium ion, something like this. But the media like this one, tryptic soy agar. The tryptic soy agar is non-synthetic because well, even you know sodium chloride 5 grams, but there are some sodium chloride contained in casein digest or soybean. So you don't know the right amount of sodium ion or chloride ion, and you have no idea how many chemicals contained in protein, casein, and soybean digest. If we classify media by purpose of use, okay, we probably use divide media to be 5-6 different types. Enrichment media, this is important because when you take sample or specimen from patient, you probably need to enrich those things, enrich the target bacteria in the enrichment media, because target bacteria are so few in the patient, but you need to enrichment before. Selective media is refer to media that contain some inhibitor of some bacteria and let the bacteria that you want to see can grow, but inhibit the other bacteria that you don't want to see. That's selective media. Differential media allow wanted and unwanted bacteria grow, but show up in different shape, different form, different color, or different appearance. Okay, lots of time, selective media and differential media may be the same media. So these media can function as selective and function as differential. Okay, minimal media is media that contain just minimum. Okay, sometimes minimal media can be selective media because the other bacteria that need so many nutrients cannot grow in minimal media. Transport media is media that can keep specimen during transfer from ward to the laboratory, or from site to the hospital, to the lab in hospital. Okay, and the last one is preserving media. Well, when you want to keep these bacteria for research purpose or want to keep that for reference, but you need to keep in a very cold freezer or liquid nitrogen, you probably need preserving media. That's the purpose. Let's say the media, the very important media that involving with gram-negative bacteria quite a lot is triple sugar iron, or TSI agar. But TSI, we not prepare TSI in a plate, but we make slant in a tube. Okay, slant is look like this area. Okay, when bacteria grow, you probably streak bacteria on slant. So bacteria exposed directly to oxygen. So this area, bacteria will use aerobic condition for growth. And also, you need to stab bacteria into the agar tole. When you stab, the technique is you not stab until the bottom of tube. You will pierce the needle around 2/3 of the depth. Otherwise, bacteria can grow out of the stab area, and that when they go out, grow by the edge of agar around the tube. That also generate aerobic condition. But if you step inside the agar, they generate an aerobic condition. So the slant and the bud have two conditions of growth. Okay, this triple sugar means they put dextrose. Dextrose is glucose polymer. When bacteria digest dextrose by whatever enzyme, dextrose hydrolysis enzyme, they generate glucose. But we put dextrose as 1 gram, so few, and put a lot of lactose and sucrose for 10 grams each. So bacteria can grow by glucose. All bacteria prefer glucose. Most of them bacteria prefer glucose. They use just 1 gram glucose quite quick. And then they need to use lactose or sucrose or both, because they contain like totally 20 grams, a lot in the media. But if they cannot ferment lactose, cannot ferment sucrose, they probably find that the other energy source from peptic digest of animal tissue or pancreatic digest of casein. These are amino acid salts, also 20 grams. Okay. So energy source can be sugar, 3 different sugar, or amino acid. And also in this media, we put ferrous ammonium sulfate and chloride thiosulfate. So these thiosulfate and ammonium sulfate, when they ferrous, when they meet sulfur, they can generate the black color. So this media also detect, can tell like these bacteria has ability to generate hydrogen sulfide or not. And we use phenol rate for to be indicator to read the result. Well, when you use needle pick bacteria, you probably stab in the agatol first, and then use the needle streak on the slants and incubate. Okay, the way to read the result. This is general TSI. Before use, the colors look like pinkish orange or something like pale red color. But if bacteria grow on slants and change the color to yellow, we call acid slant. And if the agatol or the bud change to color, from red to yellow, we also call acid slant acid bud because of yellow at slant, yellow at bud, or A/A, AA. That means glucose and sucrose and lactose were ferment and generate so many acid, and those acid convert the pH, the phenol rate indicator, to yellow in aerobic and in anaerobic conditions. Okay, this is very good for facultative and aerobic bacteria. But if they can use glucose only, they cannot use sucrose and lactose. Okay, use glucose, then they change the color from red to yellow. Okay, in the beginning they look like acid slant acid bud, yellow and yellow, but later on the aerobic condition consume all glucose because of 1 gram. Then they move to consume amino acid and generate a lot of ammonia. Ammonia turned alkaline, then the color turned back to the red. We call alkali slant acid bud, or Ka. Okay, that's referred to Shigella salmonella bacteria, and AA is mostly Escherichia or E. coli. So you know E. coli, Klebsiella, or Enterobacter. If the tube is exactly with the control, but you need to observe the streak, you probably find some bacteria grow on the streak line. We call alkaline slant alkali bud because they cannot ferment any sugar. Okay, but they consume amino acid instead in both aerobic and anaerobic. Okay, they are pseudomonads. Okay, for medical bacteria that really important in terms of medicine, we found that there are no acid slant alkali bud, but in nature we probably can find something like this. If you observe acid slant alkali bud, that because you did something wrong in the inoculation. Okay, so you probably repeat the experiment again if you see AK. Okay, let's start the bacteria from the first family, the Enterobacteriaceae. Enterobacteriaceae is very huge family of bacteria. Okay, you probably get used to Enterobacteriaceae. General characteristic, they are gram-negative, non-spore forming bacilli, so everyone knows this. The consumption of oxygen in the pattern of facultative and aerobes. That means they prefer oxygen, but they can grow in the condition without oxygen by fermentation. Okay, most of Enterobac, not all, so many species are human flora in the gut. Okay, so the main disease they can cause is diarrhea. Okay, diarrhea illness. Okay, they can be found in animal gut, in mammal gut also. Okay, they cause disease in GI tract, gastrointestinal tract. Okay, UI tract, urinary tract, and respiratory tract. So we use biochem to differentiate. So today you learn a lot of biochem tests, and I left some biochem tests not explained to you, and we learn in the unit 7. Let you explore yourself a little bit. Most of these bacteria can ferment glucose, and the other sugar also. It depends on species of bacteria. Okay, and once they ferment glucose, they produce strong acid and often gas. Well, when you grow bacteria in the tube, you probably see the gas push media, lift media up from the tube. They are all oxidase negative. Okay, this is main characteristic of Enterobacteriaceae, oxidase negative. And very impact because lots of people die per year. 3 million people die per year, and about 4 billion infected worldwide. So almost 70% of Thai population need to be experience diarrhea in your life. These are some important genus or genera of the family Enterobacteriaceae. We not learn all, but let's let you know they are very, very common bacteria like Escherichia, Shigella, Edwardsella, Salmonella, Citrobacter, Klebsiella, Enterobacter, Hafnia also, Serratia. So this is wrong, Serratia, not Serratis. Okay, Serratia, Proteus, something important, Yersinia important. Okay, if we divide genus or genera of Enterobac to be obligate pathogen and opportunistic pathogen, they can be both. Okay, obligate pathogen, they mainly attack human by pathogenesis. They cause pathogenesis. They are Salmonella, Shigella, Yersinia, and some strain of E. coli or Escherichia, not E. coli, sorry, some strain of Escherichia. But the other Escherichia, for example, E. coli, they're opportunistic. They are good bacteria in your body, but if just in case they can cause disease with you. E. coli, if they live in the wrong place in your body, they probably cause disease. Klebsiella, particularly pneumonia, can be opportunistic infection. Enterobacter, also Serratia marcescens, Proteus, probably Proteus vulgaris, okay, Providencia, Citrobacter, they are all opportunistic. Normally, they're not caused disease by healthy person, okay. Disease from Enterobac, this we talk about in general. They can cause meningitis, some Escherichia. They can cause pneumonia. They can let infected people get sepsis. Sepsis means get bacteria in the bloodstream, okay? And then you probably get systemic infection throughout the body. Diarrhea is the main symptom of this bacteria. Okay. UTI, urinary tract infection, so can be E. coli, Escherichia, particularly E. coli, some type of E. coli, Proteus, Klebsiella, Mozzarella. The virulent factor of bacteria, so they mostly have flagella. Some species can move, some species cannot move. Flagella itself we call H antigen. Okay, and the capsule. Most of the strain and species can produce capsule we call K antigen. So they can be capsule and they also immunogenic. Particularly in except in Salmonella. Salmonella we not call K antigen, we call VI antigen. That's people who work with Salmonella prefer to use this term. They have cell envelope protein. Okay, LPS is very important in gram-negative bacteria. Remember, LPS is O antigen. LPS is lipopolysaccharide. Pili is very common also. And outer membrane protein. You probably see OMP term. Okay, bacteria in this family, they name by human name. They name by the discoverer, the scientist who discovered or the doctor who discovered bacteria, or named by human to honor him, something like that. Let's start from genus Escherichia. The name by Theodore Escherich. So this guy. Escherichia normally very short rod. Okay, the rod of Escherichia is so short. You probably see sometimes it look almost coccobacilli, but very short. There are about 4 species of Escherichia. Okay, but the most important one is E. coli. Okay, E. albertii, E. hermanii, E. vulgaris also you can find some, but E. coli is very important. For E. coli, you probably get TSI, acid acid bath, with gas. Okay, with gas. This is the key for TSI. Indo-test positive, multi-test positive, they can move. They process a lot of that flagella. Urease test negative, citrate negative. Okay. The virulent factor of E. coli, they process toxin. Well, bacteria in this group, they have endotoxin for sure because they are gram-negative, and they also process exotoxin. Exotoxin in this group of bacteria we call enterotoxin. Enterotoxin is because they release in the gut. That's why they call enterotoxin. Bacteria, E. coli process 2 different types of toxin. Not all E. coli will process this, but some process Shiga-like toxin. Some E. coli have enteroaggregative Shiga-like toxin. Some produce hemolysin to lyse red blood cells, and endotoxin is LPS. They also have capsule, ion capture ability. Sometimes they need trace element, trace ion, they can capture those ions. Okay, and this can protect bacteria from the immune response. So E. coli that cause diarrhea have so many types. I just list some important type of E. coli that can cause bacterial. Sorry. The first one is enteropathogenic E. coli, or EPEC. We call EPEC. EPEC is enteropathogenic E. coli. They associated with the diarrhea in young children or in infant. Okay. So infant baby probably have fever, nausea, vomit, and diarrhea with non-bloody stool. Okay, so diarrhea but no blood contaminated in the stool. Another group is enterotoxigenic E. coli or ETEC. This causes watery diarrhea, nausea, abdominal cramp, and you probably have low fever for a week. Okay, This can spread throughout the food, particularly water, ice. No? We particularly in developing country, they cause travelers diarrhea. So when you travel to India or American people came to Thailand first time and drink some water not clean, they probably get diarrhea. Okay. Enteroinvasive E. coli, EIEC. This one can cause disease quite similar to disease from Shigella. They invade into the intestinal epithelium, okay, in human. So normally bacteria in the group of E. coli do not invade itself, but they do something else. They release some toxin or something else, but they not penetrate itself and destroy epithelium cells. This is pattern of Shigella. So once the epithelium cells or in, the wall of intestine damage, so patient probably has blood contained in the stool. So we call bloody diarrhea. So that's enteroinvasive. Okay. So enteropathogenic, get diarrhea in infant. Enterotoxigenic, traveler diarrhea. Enteroinvasive E. coli, invade. Okay. Okay. We still have enterohemorrhagic E. coli, EHEC. They cause hemorrhagic colitis or hemorrhagic uremic syndrome in young children and very old people, particularly caused by E. coli 0157H7. This is the causative strain of E. coli 0157H7. Okay. And the last one, enteroaggregative E. coli. Okay. They related to watery diarrhea, cause vomiting, dehydrating in the infant. Okay. So causative are different. The second one, we move to genus Klebsiella. Klebsiella can be capsule and mucoid colony. When you see Klebsiella on blood agar, you see mucoid, it's look mucus. When you use the loop, touch the colony, they very sticky, okay? And gram stain, you probably observe capsule around Klebsiella, okay? That's pattern. Colony bigger than E. coli, more mucoid. Important species is Klebsiella pneumoniae, but the other Klebsiella can cause diarrhea, except this one is opportunistic infection that cause pneumonia. They process capsule, they have plasmid that exchange gene with the other genus in Enterobacteriaceae. Okay. The important thing is they resist so many antibiotics. That's make Klebsiella so serious. They process toxin. Okay, they have heat stable and heat labile exotoxin like E. coli and also endotoxin. Okay, Klebsiella cause disease in a wide spectrum. Can be pneumonia, of course, can cause UTI, septicemia, can infect wound if the wound is not so clean, and septicemia can lead to meningitis and diarrhea in newborn. Well, when we talk about E. coli and Klebsiella, we probably need to talk about coliform bacteria. The coliform bacteria refer to bacteria that contaminate in water, okay? Or contaminant come from stool. This is very important. Coliform bacteria we use as a marker for hygiene. If you check the line of food production or water production, water industry, and you found coliform in the line, that means this process of food packaging or water packaging, the product probably contaminated with stool, or some worker in the factory not have good hygiene enough or make stool contamination in the target product. Okay, we refer to E. coli, Klebsiella, Enterobacter, Citrobacter, and Hafnia, but the dominant one is Escherichia and Klebsiella. Okay. Coliform can multi and non-multi. So we probably use McConkey agar for testing coliform bacteria and use indole test, motility test, urease test, citrate, Simon citrate test for 4 or 5 of these. Actually, we have the other tests I will ask you to review. Okay. Let's start from McConkey. McConkey is actually it is selective media and differential media. Selective media for what? For gram-positive bacteria because they contain crystal violet and bisalt. Crystal and bisalt together inhibit gram-positive. So the thing can grow inside this, supposed to be gram-negative bacteria. But in the media contain lactose, okay? Lactose. If bacteria cannot ferment lactose, they show up with white colony. I didn't have color. But if they can ferment lactose, they can ferment lactose because they have what? They have lactoperon, right? So then they metabolize lactose and change the color of neutral red. It turn to the pink colony on McConkey agar. But differentiate between Klebsiella and E. coli. For E. coli, the pigment, they diffuse in the agar a lot. Okay, but Klebsiella is bigger colony, look more mucoid. Okay, on both are lactose fermenter. But some strain of E. coli not diffuse like this, then you need to use the other biochemical tests. The first one, we perform motility test. In this test, we probably need semi-solid media, and we step bacteria 2/3 of the depth of the media. Okay, so this is the step line. If you observe bacteria grow at the step line, that means bacteria cannot multi. They just grow in this area. But if they can multi, they can move everywhere in the tube. And you observe the turbid tube like this. So Klebsiella cannot multi. E. coli has flagella, so then motility test positive. Okay. So at the motility test, we probably use the same tube, and you put some drop of COVID reagent. Okay, COVID reagent is resemble to phenylalanine. So indole test, sorry, not phenylalanine, it's tryptophan. We probably want to see bacteria can produce tryptophanase or not if they digest tryptophan. COVID reagent is quite similar to tryptophan, but the different thing is one, they digest by tryptophanase. The chemical in the COVID reagent will turn red. Otherwise, the color is yellow. When you drop inside this and waiting a few seconds, you probably see up to 5 minutes, you probably see the red color happen. That means bacteria can produce tryptophanase and digest tryptophan. That means indole test positive. And in this case, we read indole negative. So E. coli get positive for indole test, Klebsiella negative. So these can differentiate E. coli and Klebsiella. Okay. Urease also. Urease just test bacteria can use urea or not. If they can use urea, they generate ammonia, like Klebsiella. They change the color from yellow to pink. If the color remain yellow, that's mean negative. E. coli negative, Klebsiella positive. You probably remember this. Simon citrate. Simon citrate is ability to use citrate as carbon source. Where's citrate? Still remember? In Krebs cycle, right? E. coli cannot use that. So the color of media remain green. But if color change from green to blue, that's mean positive because using citrate can generate a lot of ammonia and alkali condition. Klebsiella get positive, okay? But if you remember multi-indo like this is summary. You probably go back home and build yourself the table for yourself, and then fill the test that important for each bacteria in the table. E. coli and Klebsiella is totally opposite. You just remember one. You can remember another one. Okay. So from E. coli, Klebsiella, the first pair, we jump to Shigella. Shigella is named from Kiyoshi Shiga, the doctor who observed Shigella. Shigella is meat spatial media, selective media, and also differential media. Selective because this media allow just Salmonella and Shigella can grow, but the other species, just few of other species can grow on SS agar. Mostly two of these species. They are TSI alkali / acid but no gas. Okay. Shigella is Ka, no gas. When you observe colony on SS agar, you probably see the transparent colony, no color. Shigella is long, slim bacilli. Okay. Based on the antigen, Shigella divide to be group A, B, C, D, 4 groups. Okay. Shigella group A refer to Shigella dysentery. This is a causative agent of shigellosis. Shigellosis in Thai we call บิดไม่มีตัว is cause bloody diarrhea. Urease negative, motility negative. Okay. For Shigella has also stay in human. So human is main reservoir. Okay, we not found Shigella in the other animal. They contaminate in fecal oral contamination. So you would get infected of Shigella by eating contaminated food or contaminated water. Okay, and also contaminated food come from contaminated hand. So then washing hand can help a lot. Okay, this is Shigella. Another species still use same media called Salmonella. Salmonella generate black colony on SS agar. So it's different from Shigella. And the black colony actually it show up with fish eye colony. So the black is in the middle of colony, not everywhere in colony on SS agar. It's Ka with gas and hydrogen sulfide. Okay, this is hydrogen sulfide. The black color is key different from Shigella. Okay. So the name is Professor Daniel Salmon. That's why name Salmonella. Okay. You probably heard Salmonella typhi, Salmonella paratyphi, whatever. They are not species of Salmonella. Okay. They are serotype. Okay. Salmonella contain like, I'm not sure, like 1,000 of serotype. It's quite a lot. But for biological species, Salmonella has 2 species. The important species of Salmonella is Enterica. Okay. So we not call Salmonella Enterica typhi. It is quite long. Then people use Salmonella typhi, but typhi no need to be italic because it is not a species name. Okay. Motility positive, indole negative, urease negative. This is Salmonella. Okay. The virulence because they process endotoxin. This play role for Salmonella to living intracellular. Okay. They generate capsule and fimbrial. Some species, some strain process fimbrial, some strain process non-fimbrial. That's for attachment. Okay. They cause typhoid for Salmonella typhi and paratyphoid for Salmonella paratyphi. In Thai, we call pyroxat is severe diarrhea. Okay. Paratyphoid is ไข้รักษาน้อย So typhoid fever, so you probably get high fever. Unlike the other fever of diarrhea, you have low grade fever. Or some people get no fever but stomach ache. You probably get headache, fatigue, lose of power, don't want to eat, okay, chill, cough, and some people get rash on the tongue. Okay, next genus is Proteus. Proteus, named from the Greek god Poseidon. The Greek of Poseidon is Proteus, right? That's genus Proteus because the bacteria under gram stain is pleomorphic. Pleomorphic is means so many shapes. Okay. Same with Proteus can transform himself to so many animals. So that's pleomorphic. They basically move by swarming. When you pick bacteria in the middle of the plate, bacteria can grow in radius and form the film-like pattern. When they move and they stop moving a little bit and then move and stop moving, that's why you see the wavy of swarming. We call swarming phenomenon. Only Proteus vulgaris can do this. Okay, on blood agar, on nutrient agar, whatever, you observe it very easy. Proteus cause food poisoning and also urinary tract infection. Proteus culture of Proteus smell fishy, bad smell quite bad when you incubate that. They cause a broad spectrum syndrome also, UTI, and also diarrhea until blah 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 septicemia. Oh, and one thing is acute otitis media. This is also important for Proteus vulgaris. So we not point you to remember all, but just know important symptom. When we talk about this keyword, you probably think about which bacteria. Genus Serratia is named from Serafino serrati. Okay, so TSI, acid acid bath, or some species of Serratia get alkaline slant acid baths. With or without gas, that's okay. Non-lactose fermenter, chemotide, okay, citrate negative, okay. It can be found to be respiratory tract infection and urinary tract infection. The significant thing of Serratia is they resist to so many antibiotics. Okay, that's make Serratius very important bacteria. Okay, so we move to next family. Oh, before we move up to the next family, let I remind you, family Enterobacteriaceae oxidase negative in all family. Okay, in all family. Then we move to Vibrionaceae. Vibrionaceae is bacilli, but look curvy. Okay, and contaminate with water. Water is main contamination, even fresh and marine water. Okay. So they mostly gram-negative curve rod, motile, bipolar flagella. They are very common in water. They live in water, both fresh marine or estuarine. Estuarine is in between fresh and marine water. Mm-hmm. And cause diarrhea. The three genera that cause disease in human is Vibrio, Campylobacter, and Helicobacter. This group is oxidase positive, differ from Enterobac. So let's start from very important one, Vibrio chlorella. Vibrio chlorella needs spatial media. This is again thiosulfate citrate by salt sucrose or TCBS agar. TCBS agar is normally green. They contain so many inhibitor for most of bacteria. Okay, V. chlorella grow. The important one of this is they ferment sucrose, not glucose. They ferment sucrose, okay, and they form acid, and they change the color from green to yellow. Okay, that's the thing. You probably see yellow colony and also yellow clear zone. Okay, that's Vibrio chlorella. They process H antigen of flagella, okay, and O antigen, okay, of somatic polysaccharide. And Vibrio chlorella also can have antigenic variation because they change antigen itself to escape immune response. Okay, to differentiate Vibrio chlorella, we have toxigenic Vibrio chlorella and non-toxigenic Vibrio chlorella. Okay, we basically all antigen can divide to be more than 100 or 140 serotype. We call O1, O2, O3, O4 until O140. The very important one is Vibrio O1 and Vibrio O139. That's important bacteria. But the other antigen is harmless Vibrio, okay? O139, sometimes we call Bengal strain, okay? This cause epidemic chlorella. Chlorella, actually, the name of disease that V. chlorella cause. Chlorella in Thai we call Ahiwa Takaro. So in toxigenic Vibrio, we have O1 and O139. Okay, particularly O1 can have two different biotypes. We call O1 classical, or O1 alto. Okay, alto type or classical type, and each classical and L-tor still have serotype that's smaller than biotype, can be Inaba, Ogawa, or Nikojima. Then we call Vibrio Chlorella 01 classical Inaba, something like this, or Vibrio Chlorella 01 classical Ogawa. And each type, each serotype still have antigen A, B, except the Nikojima has A, B, and C. So L-tor has the same pattern. Okay, there are so many typing of Vibrio, but just 01, 0139 is important. We get the L-tor from Egypt, right? The 6 pilgrim patient came back from Mecca, got Vibrio L-tor type. We name L-tor because this situation happened in L-tor, Egypt. Okay. Vibrio chlorella process chlorella toxin. It is enterotoxin of chlorella. It is AB type toxin. You still remember the term AB type? Similar to Corynebacterium diphtheriae, that's AB type. Similar to Clostridium botulinum, similar to tetanus, that's AB type. Because subunit A surrounded by subunit B, okay? Then subunit B bind together with ganglioside receptor, and then let subunit A pass through the membrane into the epithelial of the intestine. Subunit A will catalyze ADP ribosylation and ADPR ribosylation, activate adenylate cyclase. So AC, sorry, this is not AD, it's supposed to be AC. And the adenylate cyclase can increase the efflux of sodium, potassium, water, and so many iron. Actually, carbonate iron also. When the iron move into the lumen of small intestine, water will move together with the iron. Then patient get severe watery diarrhea, okay? And key symptom is rice water stool. This is the color of stool. They have no brown color because everything in the intestine will be flushed off. Okay, that because the hygienic of disposal, that means stool of the patient contaminate to the other area. So then once someone get infected with Vibrio, they probably spread to the other person in the village, in the area that's far from good hygiene. Okay. So another thing that make disease severe is the supply water. You probably need to keep supply water in the clean area, in farmers area of poor country. Probably use water from river. That is not a good idea. Okay, and contamination, food contamination in fruit, in raw vegetable. Okay, so that's chlorella, Vibrio chlorella. Vibrio chlorella contaminate in fresh water or maybe estuarine. Estuarine is in between fresh and salt water. That's Vibrio chlorella. Then we move to another bacteria, Vibrio palahemolyticus. The palahemolyticus is another group of Vibrio, but this is quite opposite to chlorella. In TCBS, palahemolyticus generate green colony. They live in the marine condition, halophilic bacteria. They love, they tolerate a lot of salt. Okay, um, these bacteria cause acute gastroenteritis. They live in fish, in shell, in squid, in so many seafood. Incubation is very quick, can be 5 hours or up to 72 hours. Okay, so in some case that you probably go to travel and eat seafood, and seafood get contaminated with, that's not fresh seafood. If you leave seafood for a while, so Vibrio parahemolyticus start growing and contaminate, and they produce some toxin, and then you get sick from those bacteria. You probably get nausea and vomiting a lot. Maybe low fever, unlike the Chlorella. Chlorella is high fever. You get watery diarrhea, or in severe case, probably get bloody diarrhea. To differentiate between Chlorella and parahemolyticus, well, first thing is TCBS. TCBS show up yellow or green, okay? Another one is salt tolerant taste. We probably culture unknown bacteria or Vibrio in 0% sodium chloride, 1% or 8%. If you see it's turbid in 8%, that means they prefer to grow in high salt. Vibrio parahemolyticus will not grow in 0% sodium chloride. They will not grow in fresh water. Okay, but Chlorella can grow in 0% or 1%, but not 8% sodium chloride. Okay. Campylobacter jejuni. Campylobacter jejuni, the cell is quite twist. If we not know indeed, we probably think like this is spirochete, but it is not spirochete. It is comma, or sometimes we call S shape or gull wing. When you see the gull try to open the wing, they can multi by flagella. They love microallophilic. Campylobacter jejuni contaminate by animal. Okay, they pathogen in various animal. Okay, they contaminate in food, and well, in the food packaging or something that contain complete package, seals perfectly, no oxygen or very low oxygen, that allow Campylobacter jejuni grow, is because it's microallophilic. Okay, this is not, well, we also saw some cases in Thailand, but most of the cases are in wealthy country. Okay, not our country, right? Not wealthy enough. So in Europe, in UK or in the US, people go to supermarket and shopping food for the family once a week. Probably they go to buy a package in supermarket that's sealed and form a microallophilic condition. But in case that Thai people go to buy food directly from the shop, or if you go to fresh market, not supermarket, to the regular market in Thailand, we cut pork or chicken, we not seal, like vacuum seal, something like that. So chance to get Campylobacter jejuni outbreak is lower than in Europe or America. The last one, Helicobacter pylori. This bacteria is spiral shape. It's little curvy. The cell is quite long, okay? Longer than Campi, okay? The Helicobacter can tolerate acid in the gut, in stomach. The optimum pH for Helicobacter is 6 to 7, pH 6 to 7. This is because in the inner membrane of the inner wall of the gut, actually epithelial cells produce mucus layer. It's quite thick mucus layer, and Helicobacter pylori move inside the mucus layer and grow inside this. This make immune cells cannot capture Helicobacter pylori. So we knew Helicobacter is a causative agent of gastric ulcer. In the old days, people thought that gastric ulcer because you release of the gastric enzyme, and the gastric enzyme or low pH substance digest your gut because of yourself, the item inside your body destroy your body actually. But later on, when we know that this action of Helicobacter pylori, antibiotic can kill the gastric ulcer, it can contaminate to the other area if they move deeper, okay? That's Helicobacter pylori. So these are all of the family. Just let you know the superficial idea how they important, how they differentiate each other, and the lab that help them differ. Okay?